Dear students, welcome to the UGC lecture series on botany. The topic for today's lecture is fossil gymnosperms, a general account. Let us first introduce the topic. The gymnosperms are the most ancient seed plants that originated during the Paleozoic era but flourished well during the Mesozoic. The fossil seed ferns or cycadophyllocales suggest the origin of gymnosperms from an ancient stock of fern-like individuals. The extinct gymnosperms are included under four orders, namely 1. Pteridospermales, 2. Bennettite tails, 3. Pentoxylales, and 4. Cordiatales. Let us take up the first order, that is Pteridospermales. It is also called cycadophyllocales. The Pteridospermales or seed ferns came into existence during the Upper Devonian and lived through Carboniferous and Permian periods and reached their climax in the Mesozoic era. Pteridospermales in India occur in the lower, middle and upper Gondwana systems that were formed in the Paleozoic and Mesozoic eras. The plants of this group had erect, slender or weak stems which bore large, pinnately compound frond-like leaves with resistant cuticle. The microsporangia had no annulus and were grouped into synangia. Pollen grains were monolete or trilete and lacked pollen tubes upon germination. The ovules were borne on modified or unmodified foliage. These ovules were cupulate, had integuments, either free or fused with nucellus. They also had a distinct pollen chamber and a micropyle. The seeds resembled those of recent cycads. Sporn in 1965 divided the Pteridospermales into 7 families, namely Lygenopteridaceae, Medullosacea, Calamopteaceae, Glossopteridaceae, Peltospermaceae, Corystospermaceae, and Catonaceae. The first 3 families were known only from the Paleozoic era, whereas the rest have been reported from the Mesozoic. Lygenopteridaceae was represented both in the upper and the lower Carboniferous periods of the Paleozoic era. Medullosaceae, named after the type genus Medullosa, are found in great abundance in the upper Carboniferous and the Permian rocks. The family Calamopteaceae is named after the stem genus Calamoptes, discovered from the Thuringian strata of the Upper Devonian age in the form of petrifications. Glossopteridaceae flourished well on the Gondwana land during the Gondwana Ice Age. The family Peltospermaceae includes form genera Lepidopteris for leaves or fronds, Peltospermum for seed-bearing organs, and Antiocia for pollen-bearing organs. These were collected from the late Triassic. The family Corystospermaceae was constituted by Thomas in 1933 to include some fossils collected from the Triassic plant beds. Catonaceae is a small group of compressions of controversial plants that were first described by Thomas in 1925 from the Middle Jurassic plant beds at Caton Bay in Yorkshire. These extinct plants range from the Upper Triassic to the Upper Cretaceous. Let us look into the characteristic features of families of Pteridospermales. We will first take up the stem. In Lygenopteridaceae, stems were either climbers or had a straggling growth habit. The genus Medullosa has been reconstructed as a tree-like form. The stems were unbranched and varied in length and diameter in various species. In Calamopteaceae, the type species was named as Cytinomylontedianum. It was found in the form of petrified pieces of a flattened stem. The Glossopteridaceae include Vertebraria as the organ genus, representing the stem, usually fossilized in the form of flattened, branched or unbranched axis, which in some specimens bear lateral, slender and branched roots. The stem fragments of Peltospermaceae and Corystospermaceae have not been fully described. The stem in Catonaceae is a small, branched twig bearing foliar bed scales and a C-shaped leaf trace. It indicates that the plants were either branched trees or shrubs and not thick, stumpy or columnar and unbranched. The leaves were compound circinate, and the ultimate leaf units dissected in Gymnosperms. Neuropteris is the common leaf type found in association with the various medullosome types of stems. In Neuropteris, the fronds are of considerable size, measuring up to 10 meters and are bi- or tripinnate. The racks may be dichotomously branched, and both racks and petiole bear pinnules. The pinnules are mostly entire, rarely slightly lobed, and broadly linear with cordate base. The incomplete fragments of leaves found in association with these stem pieces do not reveal the structure clearly in Calomopteaceae. However, leaves in Glossopteridaceae were spatulate and tongue-shaped. These leaves were simple, had a distinct midrib and reticulate venation with entire margin. In Peltospermaceae, Lepidopteris is the generic name given to the sterile and bipinnate leaves that were 8 to 30 centimeters long and lanceolate in shape. Each leaf or frond had a stout rax which is unforked and bears pinna on either side. The pinna bear pinnules that were closely set and linear or lanceolate in shape. Corystospermaceae include the form genera Dicrodium and Xylopteris for leaves or fronds. The frond had a dichotomously forked rax and was unipinnate with sessile pinna attached by their broad bases. In Catonaceae, the leaf consisted of a cylinder petiole bearing 4 terminal leaflets. Each leaflet consisted of a distinct midrib that gives off finer branches that fork freely, forming a network of veins. The leaflet or even the whole leaf fell by the formation of an abscission layer, which is an angiospermous character. Let us take up the reproductive organs of families of pteridospermales. We will first discuss male reproductive organs. In Gymnosperms, microsporangia were born on the ultimate branches of the frond, which occur in paired synangia. Each microsporangium was about 3 millimeters long and bilocular. Each loculus was full of several trilete microspores. The genus Vitellacea is a microsporangiate organ of the family Medullosaceae. It was a hollow and bell-shaped structure with a single ring of tubular microsporangia on the wall that were concrescent. Both male and female reproductive structures are not known in Calomopteaceae. Arborilla vulgaris was the sporangia-bearing organ of Glossopteridaceae. They were stalked sporangia, more or less oval in shape, and contained 2 winged spores. In Peltospermaceae, the pollen-bearing organs are put within the genus Anteucia, which is a pinnately branched structure. Each secondary branch bore up to 12 pollen sacs or microsporangia. The microspores were wingless and oval in shape and had a single longitudinal furrow on the outer wall. The male reproductive organs are grouped into the form genera Pterococcus and Pterorax in Corystospermaceae. The fructification consisted of a central axis from which arose short lateral branches. Each lateral branch terminated into a peltate head, which bore up to 30 microsporangia on the underside. Microsporophyll or Catonanthus in Catonaceae consisted of a dorsiventral rax, which was pinnately branched. The ultimate branches bore synangia on their surface, and each synangium was radially symmetrical with 4 pollen sacs. Female reproductive organs. The ovule in family Gymnosperms was barrel-shaped, radially symmetrical, with a single stout integument, which consisted of an external hard and stony sclerotesta and an inner fleshy endotesta. External to the integument, the ovule was partially enclosed by a lobed cupule. The medullosome seeds were derived from an ovule with a single massive integument, which is distinguished into 2 seed coats, namely outer fleshy sarcotesta and an inner hard sclerotesta. In Glossopteridaceae, the female reproductive organs consisted of a cupulate and multiovulate fructifications. In the former, the seeds were protected by cup-like or circular disc-like structure called the cupule. Each cupule contained only 1 seed. The cupules were borne terminally on each pedicel. Multiovulate fructifications possessed bilateral and biconvex cushion-like receptacles borne singly on long shoot or short pedicels. The receptacles bore numerous ovules or seeds which were embedded in the depressions. In Peltospermaceae, the seed-bearing axis was cylinder, which bore superiorly arranged branches that terminated into peltate heads, each bearing a number of oval seeds on their lower side near the margin. These seed-bearing axes are put under the genus Peltospermum. The female reproductive organs were described under 3 seed-bearing genera in family Corystospermaceae. These are Oomacomacea, Philophosperma, and Spermatocoda. In all the 3 genera, the seeds were enclosed within cupules, which were helmet-shaped in Oomacomacea and Philophosperma, and bell-shaped in Spermatocoda. The fruit genus in Catonaceae is Catonia gnathostae. Catonia consisted of a dorsiventral rax bearing a number of lateral stalked outgrowths, swollen at their tips. These outgrowths were called fruits or cupules. The fruit, when removed from the rax, consists of a stalk which broadened terminally into a pouch-like structure. The seeds were pendulous, orthotropous, ovoid, and oval or flattened. The second order of fossil gymnosperms is Benedictales. It is also called Cycadioidales. This group flourished well during the Triassic to Cretaceous periods of the Mesozoic era and became fossilized either in the form of compressions or petrifications. Except for the root, rest of the plant organs have been discovered in a petrified form. Bucklandia was the first member of this group that was collected and named in 1825. They are entirely extinct and resemble cycads in outward stumpy appearance of the trunk and the apical crown of large pinnate compound leaves. The petrified stems of this order reveal them to be upright, columnar, conical, and less than a meter in height. They grew very slowly. The leaves are large and pinnately compound. Venation was unicostate parallel and stomata were syndetochelic. The reproductive organs or flowers were bisexual in majority of the genera, but unisexual condition was not rare. Flowers arose in axils of leaves in all the Benedictales and resembled in their general orientation to the flowers of genus Magnolia among dicotyledonous angiosperms. The pollen grains were borne in sessile or stalked bilocular synangia. Stalked or sessile cupuleless ovules were interspersed by interseminal scales on the receptacle. Sporn in 1965 divided Benedictales into 3 families, namely Cycadioidaceae, Williamsoniaceae, and Villandilaceae. Cycadioidea antrusca, belonging to family Cycadioidaceae, is the oldest fossil ever collected by man. Williamsoniaceae includes the genus Williamsonia, discovered by Williamson. He named it Zamia gigas, but later its name was changed to Williamsonia gigas. Let us discuss the characteristic features of families of Benedictales. Stem. Cycadioidea had a short, branched or unbranched, spherical to conical to irregular trunk, marked by several rhomboidal leaf bases, with multicellular hairs in between. The stem in Williamsoniaceae was branched, slender, and columnar, covered with armor of rhomboidal leaf bases. The branching was lateral and projected through the armor of leaf bases and was of 2 kinds: A. Sterile or vegetative branches, B. Fertile shoots. The stem was much branched and about 1.5 meter tall in Williamsonilla coronata in family Villandilaceae. Let us take up the leaf of Benedictales. The trunk bore a crown of pinnately compound leaves, about 10 feet long in some specimens of Cycadioidaceae. In Williamsoniaceae, at the summit of trunk was a crown of pinnately compound leaves named as Tylophyllum cucens. The leaves were of 2 types: A. Simple scale-like and pointed leaves, B. Pinnately compound foliage leaves. In Villandilaceae, the leaves, named as Nelsoniopteris in Williamsonilla coronata, were simple, entire, linear or lanceolate to narrowly ovate and petiolate. Reproductive organs of Benedictales. Flowers in majority of species in Cycadioidea, that is family Cycadioideaceae, were bisexual and arose on short stalks, singly in the axil of each leaf. Williamsonia, belonging to family Williamsoniaceae, was unisexual and dioecious. The flowers were borne terminally in leaf axils on lateral shoots. In Williamsonia coronata, belonging to family Villandelaceae, it had hermaphrodite flowers. Each flower bore a number of sterile parient lobes densely covered with long hairs. Microsporophyll or androecium. The microsporophyll or androecium consisted of about 20 pinnate appendages arranged in whorls and were united by their base to form a disc in Cycadioidea. The fertile region of the microsporophyll bore tuberculi that connect the outer wall of the microsporophyll with the inner, and along these tuberculi were borne the synangia or pollen capsules. In Williamsoniaceae, each male flower was surrounded by a series of parent-like bracts and consisted of whorls of varying number of microsporophylls united at the base to form a cup-like structure. Generally, each microsporophyll had a sterile, stalk-like portion and upper fertile part, bearing 2 rows of finger-like synangia. Each synangium contained 2 rows of chambers enclosing microspores. In Williamsonia coronata, there was a ring of 12 to 14 microsporophylls that were free from each other and attached to the base of receptacle or the central column. Each fleshy microsporophyll appeared like a pip of an orange fruit and had finger-like outgrowths arising from its middle portion, enclosing synangia. Female flower or gynoecium. It consisted of a conical or spherical receptacle that bore numerous stalked ovules and interseminal scales in Cycadioidea. The fused tips of interseminal scales formed a sort of external protective covering or pericarp around seeds. In Williamsoniaceae, each flower had a distinct receptacle surrounded by parent-like bracts, which had scales at their base and hair-like ramenta above. The stalked ovules and interseminal scales were arranged in closed spirals around the conical receptacle. The gynoecium in both the genera of family Villandelaceae consisted of a central broad-based conical receptacle that tapers to a narrow and capitate sterile tip. In Williamsonia coronata, about 300 stalked ovules and about 1,200 interseminal scales were spirally arranged around the central column. The third order of fossil gymnosperms is pentoxylales. It was named by Professor Birbal Sahani in 1948 after the type genus Pentoxylon. These plants have been treated under a separate order by Sporn in 1965 and have been found to occur in silicified petrifications. The order includes a number of form genera. For example, Pentoxylon and Nipanioxylon representing stem, Nipaniophyllum representing leaves, Sahania representing pollen-bearing organs, and Cornoconites representing seed-bearing organs. The members of this group possess a unique combination of characters of Benititales, Cycadales, and Coniferales. But a few characters are peculiar to the group itself and hence deserves a position as a separate order of the Cycadopsida. Characteristic features of pentoxylales. Let us discuss stem first. Plants of this group were probably shrubs or small trees. The branches were of 2 types, long shoots and dwarf shoots. The long shoots exhibits prominent armor of rhomboidal leaf bases and spirally arranged simple leaves. Short lateral shoots had spirally arranged leaves and also bore reproductive organs terminally. Reproductive organs of pentoxylales. Reproductive organs were unisexual. Let us discuss the male fructification. Sahania nipaniensis, the microsporangiate organs of the pentoxylon, were terminal on lateral dwarf shoots. Each consisted of a dome-shaped receptacle with about 24 microsporangiophores arranged around it in a single whorl. Each sporangiophore bore spirally arranged branches. These branches bore terminal pear-shaped microsporangia. The sporangia were unilocular and contained several boat-shaped and monolith pollen grains. Female fructification. The peduncle of the infructescence was divided into several branches or pedicels. Each pedicel terminated into a female strobilus, and each had a central receptacle to which was attached about 20 sessile ovules. The ovules were surrounded by a single integument that was free from the nucellus. The integument had an outer fleshy sarcotesta and an inner stony sclerotesta. The last order of fossil gymnosperms is chordatails. It represents the most ancient and primitive order of the class Coniferopsida. Plants in this order appeared in the Devonian and were at their peak of development during the Carboniferous period of the Paleozoic era. This fascinating group of gymnosperms was named after the 19th century paleobotanist called Corda. The plants belonging to this order were tall trees with slender and monopodially branched stems. The leaves were simple, subulate, spirally arranged with parallel venation. The strobili were unisexual and compound structures, which consisted of a central axis that in turn bore fertile shoots bearing sterile and fertile appendages. The order has been divided into three families, namely Chordataceae, Aristophytaceae, and Poroxylaceae. Let's discuss the characteristic features of families of chordatails. The members of Chordataceae formed the world's first great forests towards the close of Lower Carboniferous and included tall trees that grew in dense stands. The family Poroxylaceae is pre-Carboniferous in origin and is represented by a single genus Poroxylon, whose seeds are put in form genus Rhabdospermum. However, the family Aristophytaceae includes three form genera, namely Aristophyton, Endoxylon, and Beringia. Stem of chordatails. The stem was slender and about 30 meters tall and attained a diameter of about 65 centimeters. The branching was lateral, confined to the top of the tree. The chordatalian stems have been described under several generic names. All of them, however, agree to the anatomy of chordates and mesoxylon. Leaf of chordatails. The leaves showed considerable variation in family Chordataceae and were simple, subulate, lanceolate, spatulate with parallel venation. In Poroxylaceae, the leaves were simple and up to 1 meter long. Reproductive organs. The fructifications of chordates are included under the form genus Chordatanthus. The fructifications were loose inflorescences or compound strobili and are borne among leaves. Each consisted of an axis bearing spirally arranged bracts with axillary reproductive shoots. The compound strobili were unisexual. We will first take up the male strobilus. It is also known as microsporangiate short shoot and consisted of a central axis with a large number of spirally arranged scales. The proximal scales were sterile and distal fertile ones possessed 4, 6 or more finger-like microsporangia or pollen sacs. The microspores had an equatorial bladder or an air cavity called saccus enclosed by the exine. Female strobilus. They arise as lateral outgrowths from the inflorescence axis and have each a central axis with spirally arranged appendages. The distal fertile ones bore 1 to 3 ovules terminally, each enclosed within a single but parted integument. The integument is distinguishable into outer fleshy sarcotesta, middle sclerotesta, and inner narrow and fleshy endotesta. Dear students, that is all for the present lecture. But before concluding, let's refresh few points which we actually learned during the course of present lecture. We came to know what are fossil gymnosperms. We also understood the morphological and reproductive features of various families of pteridosperm males. Also, we highlighted the vegetative and reproductive characters of bennettitails. And lastly, we were able to discern the characteristic features of pentoxylales and chordatails. Thank you.